introduce everyone to our program, and look at all the opportunities we have at pre-health shadowing if you have not previously attended our shadowing sessions. So pre-health shadowing is a student-led, minority-led, women-led nonprofit dedicated to helping prospective healthcare professionals gain access to educational resources, no matter their demographic status, abilities, or location. So my name is Muntaha. I am the editor-in-chief at Pre-Health Shadowing, and thank you all for attending our session today. Let's get started. So just a little PSA, we do have closed captioning for all of our sessions to accommodate all students. This setting is available on the bottom of your Zoom screen. If you need assistance enabling the transcript, please direct message one of our team members. We are always looking for ways to be more inclusive and ensure our sessions are accessible to everyone. So please, if you have any recommendations, make sure to email us at info@prehealthshadowing.com. So since this is an international program, we do want to know where everyone is Zooming from today. So drop in the chat where you're calling from. It's great. We have people from New York and Los Angeles. Nice. So if you want to keep in the loop, follow us on social media. We are active on Instagram, TikTok, or you can sign up for our email list on our website so you can never miss a session. We have some wonderful opportunities for you all as benefits being part of the program. We have partnered with Kaplan to get our students a 10% discount code that can be used on all Kaplan products, as well as free resources such as study guides to help prepare you for standardized tests like the MCAT, NCLEX, or PCLAT. If you fill out our short survey in the chat, we will get in signed up for these deals for free. We would also like you to draw your attention to another amazing program here on the slide called Neolith. Neolith is an online mental health platform for students. College and young adulthood is stressful enough as it is in non-pandemic times. So for pre-health professionals especially, we know the path isn't easy. That's why we have partnered with Neolith to spread the word and offer free access to their services. If you use the link given in the chat or enter the code PREHEALTH when signing up, you have access to their services. So the next slide, we'll be talking about Mask for Masks, which is an amazing women-led organization that donates 4 masks for every 4 masks sold. These go to people in need during the COVID-19 pandemic. Those in the homeless community, healthcare workers without proper PPE, and others who are struggling to stay safe. When our discount code PHS15 on the screen there, you can get a 15% off your order. If you buy through this method, Shadowing will also get 10% of the proceeds, which is amazing because we are a nonprofit that runs solely off the support of our community. If you want to play a bigger part in supporting PHS, we would love for you to join our network of student volunteers and team members. You can apply to be part of our administrative team and lead students in various projects and initiatives with professional outreach, grant writing, and more. We understand that as a pre-health student, you may not have the time, so we also offer the opportunity to volunteer asynchronously with tasks that can be done on your own time. We would love to have you be a part of this program and contribute your own unique perspectives and experiences. If you are a high school student and want to get involved, we have started a program called HTP, High School Training for PHS, which allows you to connect with colleges and pre-health programs and get involved in fundraising for PHS and organize resources for other high school students who might be interested in medicine and the health field. We want to recognize the hard work of all of our students in the program. So if you are interested in getting published, you can submit essays, reflections, research papers, and reviews to me, I'm the editor-in-chief, through the link dropped in the chat to have your work on our website. This will look great on CVs, applications, resumes, etc. So please take advantage of this, and I do look forward to reading any submissions. You can even reflect on today's session and submit one. Part of our mission at PHS is to promote diversity. In order to do this, we have launched an initiative to have monthly panels to celebrate different demographics in the field of medicine. Some of these upcoming events include a series on patient experiences, a COVID-19 roundtable, international student forum. If you have a mentor, professor, or professional that has inspired you and you think you could contribute to these conversations, please nominate them today using the link in the chat. If you can, we do humbly ask that you donate to our program. As you know, pre-health shadowing is completely student-run, and we are working around the clock to keep this free and accessible to everyone. So unfortunately, Zoom and our website are not free, so any contribution you can give would be greatly appreciated. If you are not financially able, we request you send this link in the chat to someone you think can contribute to the donation. Throughout the session today, we encourage you to drop any questions you have for the speaker in the chat. Our team members will be making note of these, and we will be asking them to our health professional at the latter half of the session. Make sure to take good notes as our professional is going over their presentation, as there will be the chance to take a post-shadowing assessment to verify your virtual shadowing hours. More information will be available on this at the end of the session, so please stay tuned. Lastly, if you can, we request you to turn your cameras on. This is by no means an obligation, but it does help to feel closer in this time when we're all socially distancing. I appreciate you for listening, and now I would like to welcome Dr. Barrett. Thank you so much for joining us today. If you may share your screen whenever you are ready. Dr. Barrett, you can unmute yourself.
Of course, there has to be at least one time during every Zoom meeting where somebody starts talking without unmuting themselves first. So thank you so much for the warm welcome. I'm really pleased and privileged to be here with you this evening or this afternoon or wherever you happen to be. I even saw somebody as far afield as Nigeria popping up in the chat. So it's exciting to have the opportunity to talk to people in so many different places. I am a professor in the School of Medicine at the University of California, San Diego. I have been there for, 36 years almost at this point. So essentially my entire professional career. And I am a PhD, not an MD, but I have always worked in the setting with clinicians, clinical colleagues, people involved in medical research. And my title, A Career in Diarrhea, doesn't necessarily usually spark huge amounts of enthusiasm, but I'm hopeful that I can convince you by the end of my presentation that this is not only a really interesting topic to work on, but also one that's really important. I'm also spent some time talking about my path to my career and the sorts of things I get involved in during my time at work. I found this picture online, but it's intriguing to me because part of what I have done is worked on mouse models of diarrhea. So I tell my husband, who's not a scientist, that I spend my time giving mice diarrhea. Unfortunately, this picture is not very anatomically correct, but because with his tail in the front there, he wouldn't really be able to poop. But besides, it's a cute image, and I sort of collect pictures like this. Slightly strange thing to do. Okay, now this worked before, so why isn't it working now? Okay, for some reason it is not advancing. So let me try.
Are you clicking on another screen, perhaps?
I'm just clicking the buttons, the arrow buttons, and it's not advancing, and it was before. So let me try stopping sharing and sharing again, unless you have another idea, Hannah.
I was just going to say, if you can click on the screen, use the buttons.
Oh, no, that's not doing it. Oh, yes, there we go. Now I've gone too far. Okay. So as you hopefully can tell from my accent, even though I've been in the United States for quite a long time at this point, almost 40 years. I'm originally from the United Kingdom, and that's where I grew up and did my undergraduate and graduate work in London at University College London. And at the time I was doing my studies, there was a very strong pull for anybody who wanted to make a career in scientific research to at least spend some time in America. So that's basically where I'm from and how I got here. I was a really nerdy kid. I really liked science. I liked chemistry in particular. I mercilessly begged my parents to buy me a chemistry set. And then because I was a nerdy kid, I very systematically worked my way through every experiment in the little book that came with it. And I thought I was going to be a synthetic organic chemist. And I went off to college to be a synthetic organic chemist and realized very quickly that I wasn't actually very good at synthetic organic chemistry. But even prior to that, I had started to sort of really think about where chemistry, my first love, could be best applied. And in fact, as an undergraduate, I had chosen to major in medicinal chemistry rather than straight chemistry. And the synthetic organic chemistry part of it was that I was going to make drugs. So I thought, well, if I'm not a very good synthetic chemist, maybe I can apply my training in chemistry to really investigate problems in biology and to work in the biomedical sciences and collaborate with physicians to make advances in actual human disease. The other thing that is really important to know about me is I am the world's greatest introvert. You guys are probably old enough to actually have seen the movie The Incredibles, and this is a picture of Violet Incredible. So if you haven't seen the film, I strongly urge you to see it. Violet Incredible is noted for being painfully shy and usually has her hair in front of her face. And this was what I was like as a child also. I also at the time had an eye condition which made my eyes very teary most of the time. And so the combination of always having my hair in front of my face and having my eyes screwed up closed most of the time did not exactly make me a welcoming and open person for people to get to know. I think looking back, I kind of, I did better than I realized at the time, but I definitely felt majorly challenged in interacting with people. I was much more fond of books and thinking about science. I went to an all-girls high school, so that made me a little challenged in interacting with members of the opposite sex. And as I told you, I was a total nerd, and it's really, I think, instructive for me to realize that I have actually been able to succeed in my career, even as an introvert, and the having the ability to sort of reflect on things is an asset in science, and I've just had to figure the rest of it out as I've gone along. So I'm a really big fan of the phrase "fake it till you make it." And one of the people who really encouraged me in that was my my PhD advisor, who was also my advisor as an undergraduate. I, in fact, met him before I even went to university. The university that I attended in London had a sort of open day for, I guess, what would be high school seniors in American parlance. And at the urging of my high school chemistry teacher, I went to this open day with one of my friends from school who was also interested in chemistry. And it was so exciting. We actually spent 2 days in the department. We got to do some fairly straightforward experiments. We got to see the dorms, and we got to interact a lot with the faculty, which was really wonderful. And I was very, very impressed by this young assistant professor at the time, whose name was Fred, and he was very energetic and really inspired everybody. I couldn't imagine that anybody wouldn't want to go to that department after that. And so I pretty much decided on the spot that this was going to be my number one choice for university. It was almost a disaster, however, because on the very at the very last session we were all gathered to have tea and cookies, and Dr. Pierce, as I understood him at that point, came over to talk to me, and I literally turned around and my entire cup of tea spilled all across his front. And so I, of course, was mortified, and I thought my career was over before it was even started. And then when about 6 months later, I was invited to come and interview for a place at the university, who was my interviewer, but none other than Dr. Pierce. He was very gracious and didn't mention the spilt tea incident, although he did confess to me when I was finishing up my PhD that he did remember that. But he's just been a great mentor all my life. So the one takeaway from this is find yourself mentors, as many as you can. You need multiple mentors to advise you on different things, and find good mentors who care about you and what you want to do, and will help you get there. And then find mentors who can see beyond the limitations that you see in yourself. So I was reasonably successful during my PhD. I got some papers published, I got experiments done, but I was absolutely petrified about what the next steps might be. And as I told you, I was Violet Incredible. I didn't really want to talk to people. And Fred was the one who used a soccer analogy. So he was a lifelong fan of a soccer team in London called Arsenal. And he had season tickets, he went to all the games, and there is a soccer term to put yourself about a bit. It means to make sure you're all over the soccer field and you chasing the ball on every opportunity. And it sounded a little racy to me, but he said, you've just got to get out there, Kim, and put yourself about a bit. And so with his encouragement, I came to the United States. Even though I had never been, I'd never spent any time outside the UK. I expected to live my whole life in London. I never expected to go anywhere else. And he was the one who said, you really should consider going to the US and pursuing a postdoctoral fellowship there. It will set you up for your academic career. So that's what I did. So here is my pathway. I got my PhD in chemistry, actually biological chemistry at University College London, in London. And then I came to the NIH in Washington, DC, in Bethesda, and did my postdoc in a very much MD-focused lab. So my mentor, Dean Metcalfe, was an MD. The other trainees in the lab that I worked with were all MDs doing their fellowship training in allergy and immunology, and in some cases in ENT, because it was sort of an allergy-focused lab. That was really the first time I had the opportunity to work with people with medical and clinical training, as opposed to the basic scientists I'd worked with up till that point. And that was also a revelation for me, and really taught me that I wanted to have my career in a setting where I would be able to directly interact with people who could give me insights about how my basic science could apply to actual problems that they were seeing in their patients. As I was finishing up my postdoc at the NIH, it was actually, my intention had been to return to the UK and look for a faculty position there, but it was a very difficult time in the UK. The sort of mid 1980s, the economy in the UK was really in pretty bad shape, and the government of the day was not highly supportive of science and of academia, for that matter. And so there were really just no jobs to go back to. And out of the blue, I got a call one day from a professor at UC San Diego saying, you know, I've seen that you're listed on this list of people who are looking for postdoctoral fellowships or to look for positions. And I wonder if you might consider coming and working with me in my lab. And he was a gastroenterologist. And so that was the sort of final step, is to move into the GI field. So I didn't know him. I didn't know where San Diego was. I did find out that it was a very long way from Washington, D.C., and even further from the UK. But it just so happened that I was going to be presenting my work at a scientific meeting in California a couple of months after that time. And so we agreed that I would go and visit. Those of you who are out in California know that San Diego is a pretty beautiful place to be. And so it wasn't such a terrible place to end up. And completely on top of that, UC San Diego is a very exciting institution to work in. It's very collaborative. It was a very young institution at the time, and it had sort of lots of novel ways of doing things. So I started off there as a research scientist, and then I got what was called an in-residence faculty position when I started to get my own research funding. Eventually, I became a tenure-track faculty member, and I'm currently a distinguished professor in the Department of Medicine. And I've also been vice chair of the department, where I focused on the research activities of the department. I was responsible for advising the chair on strategic directions, helping to assign lab space, and thinking about recruitment of new faculty. But I also had the opportunity about 15 years ago now to take a campus administrative position. So I also spent 10 years as the dean of the graduate division, overseeing graduate education for all of the students across campus. So not just science or STEM students, but also in the humanities, the social sciences, people doing MD PhDs, people doing master's degrees, just the full spectrum, and that was really fun. When I stepped down at the end of my term of that position, I went back to the faculty, but just for the last 14 months or so, I've had the opportunity to do something that's also related to graduate education, because I'm currently working at the National Science Foundation, and I've kind of come full circle because I'm back in Washington, D.C., and I'm the director of the Division for Graduate Education. So some of you may be aware of the NSF Graduate Research Fellowship Program, which provides fellowship support for students pursuing masters and PhDs. We also have a number of other programs. And it's very exciting now to contribute on a national level to thoughts, ideas about how to transform the way we do graduate education, how to prepare students for a full range of career opportunities. And one of the things that's really near and dear to me, and I was really pleased to see that it's part of the sort of ethos of your organization, is how do we broaden participation and make sure that everybody, no matter what group they come from, is able to participate in the STEM workforce. So I'm going to talk a little bit about my research before I sort of wrap up with telling you a little bit about, what my day-to-day work is like and how I spend my time. So I'm the first in my, I'm a first-generation college attender. Both my parents left school at the age of 14. They never even completed high school, let alone going on to university. And in fact, none of my siblings went to university either. My younger brother, well, actually all three of them left school at 16. My sister went back after she'd been in the workforce for several years and got a degree from something in the UK called the Open University, which was sort of online education before such a thing existed. I think she took most of her classes by watching the television very early in the morning. My other sister went straight into nursing training and then went back and got a degree. And then my brother is basically a manual laborer and wasn't interested in any kind of education at all. So in my family, it was often a little challenging to sort of explain what I did. And I would have conversations with my mom, and she would say, well, explain to me again what it is you work on. And I would say, well, it doesn't really make for good cocktail party conversation because, as we've already alluded to, it is not that she goes to cocktail parties, but it's not a good topic for cocktail party conversation when you tell people that you work on diarrhea. But it's really, 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 really important. And this picture is kind of a guiding light in my life. This is a textbook photo that I have used in virtually every research presentation I've ever given because I think it's really important. So the gentleman, it's a picture that was taken in Thailand. The gentleman that's seated is a patient who recovered from an acute infection with cholera. And the two ladies behind him are the nurses that took care of him while he was acutely ill. And then all around their feet are the IV bottles, the fluid bottles that were needed to resuscitate him from this disease. So in developed countries, we tend to think of diarrheal diseases as a bit of an inconvenience. Just about everybody has had experience and knows that it's kind of miserable to have a diarrheal illness, but typically, in a day or two it resolves and you go back to your normal life. But diarrheal illnesses truly are, in many settings, definitely a cause of great morbidity, but also life-threatening. And diarrheal illnesses are amongst the leading cause of death in children under 5 worldwide. So the other thing I'll say before getting into the specifics of research is one of the things that's always been really appealing to me as being part of the sort of research enterprise is what a collaborative and incremental type of work it is. Now, obviously, there are always times when people will make major sort of step change advances. But most of the time as scientists, what we're doing is gradually building up the evidence, building on the work of others and finding our brick to put in the wall, finding our piece of the puzzle to add. But ultimately, when you look back over the arc of your career, you can see that you actually have made some really significant contributions. And together with the work of others, it's very much a collaborative team effort. Whether it's within your lab or in the scientific community more broadly, everything adds up to something very significant. So why do we care about diarrheal diseases and specifically about foodborne illnesses, which are sort of the major causes of diarrheal disease? So in terms of the sort of the social impact of these diseases, in developing countries, they significantly impede socioeconomic development. So not only are these countries already suffering from, a lack of resources, but the fact that so many people are afflicted with diarrheal illnesses has long-term impacts. And one of my former trainees who is now looking at the interplay and the crosstalk between the brain and the gut is trying to uncover mechanisms for what has been known for a long time at the clinical level, which is individuals who have repeated bouts of diarrheal illness as children have significantly impaired cognitive development. And so then you've got kind of a vicious cycle because your adult population is not has a subset of individuals who've had this history and so are not as prepared to contribute to the socioeconomic development of the country. And so then you continue to have, issues with food distribution and lack of clean water and so forth. So it would be great if we can figure out ways to interrupt that vicious cycle. Foodborne illnesses collectively have an overall burden that is comparable to other major infectious disease states, such as HIV/AIDS, malaria, and tuberculosis. I've already mentioned that these are major causes of death for children under 5, but 40% of the disease burden of foodborne illness is borne by children under 5. So this is a really significant issue. Again, contributing to sort of long-term sequelae. And most foodborne illnesses caused by infectious agents that cause diarrhea, and mostly viruses and bacteria. And then there's a lot of variability by region. Africa, Southeast Asia, certain Eastern Mediterranean countries have the highest burden, but that does not mean, as we'll go on to discuss, that we are immune in developed countries as well. We have been particularly interested in infections with Salmonella. Why do we care about Salmonella? Well, Salmonella is the leading cause of death due to foodborne illness worldwide. It is also the most burdensome foodborne hazard in terms of disability-adjusted life years, which basically is just a way of measuring the impact of the disease. It is an important pathogen in children, but also in adults, and because of that, it exerts a high economic burden, and it is relevant in all regions, as we will see. So I don't know if this is going to play, and if it doesn't matter, because no, it's not going to. Well, suffice to say, oh, there it is. Let's see. So this is just a very, very short video about a Salmonella outbreak in a developed country. And if we can't hear the sound, I'll just skip it. But do you hear the sound? Good.
I think we actually are having sound issues.
Oh. Then we'll stop.
It's kind of, I think it may be very faint or something.
That's okay. It actually doesn't really matter. All you need to remember is 206 million eggs. That's a lot of eggs, a lot of livelihood for those egg farmers. But this is like part of the course. You may remember this particular thing with the eggs being recalled, but if you don't, you probably remember that spinach was contaminated with Salmonella, or other leafy greens, or you name it. And then another example, which is shown on the, no, not that. On this slide, this was a very significant case in 2009. So this was the case of Peanut Corporation of America. And it notes here that 666 people got sick and 9 people were killed. Eventually, well over 1,000 people got sick and 11 people died. And this was also in the United States. The gentleman on the right was the CEO of this company called Peanut Corporation of America. And he actually received a sentence of 28 years in prison because his company shipped product that he knew was tainted with Salmonella. His sort of microbial quality control person sent him an email saying, I think that the peanut butter is contaminated, and he sent one line back, ship it anyway. And what made this especially bad is that Peanut Corporation of America mostly made peanut butter for nursing homes, hospitals, and daycare centers. And one thing I haven't mentioned, in addition to infants being very, very vulnerable to diarrheal illnesses, so are the elderly. So vulnerable populations can get really sick because they can get very rapidly dehydrated, and then they are, apt to succumb from diarrheal illnesses. And so this is why people died in this outbreak, and most of the people who died were in those institutional settings where not only are you dealing with vulnerable populations, but you're also dealing with people in close quarters, and so the disease can spread rapidly. So that's why we care about Salmonella. Even in developed countries, foodborne infections with Salmonella can cause disease that is serious enough to result in death, as we just saw, particularly in vulnerable populations. It's also true that antibiotic-resistant strains of Salmonella are emerging rapidly worldwide. And one of the reasons that we were really interested in this disease is that, unlike cholera, the gentleman I showed you earlier, there wasn't really a very good understanding of why this bacteria caused disease. And part of that was due to the fact that there was a lack of small animal models where you could actually study this. So that's the mouse on the toilet. The other thing that you need to know about the gut is it is a huge interface with the outside world. The intestinal tract is contiguous with the outside world, and it's often stated to have the surface area of a tennis court. So folded up inside your gut, you've got a whole tennis court of opportunity to interact with the outside world. And of course, that's important from the sort of physiological standpoint of what the intestine is there to do for us, because we use it to assimilate nutrients into the body. So it needs to be contiguous with the outside world, but it also means that it is an important portal for infections to get in. And the interface is at the level of a single layer of cells called the intestinal epithelium. And starting in the stomach and going through the whole length of the gastrointestinal tract, we just have this single layer of cells as the protection from the stuff outside and the rest of us on the inside. So the outside is the lumen, the inside of the tube, and the outside is what's called the lamina propria. That's the tissue immediately below this single cell epithelial layer. And what does this epithelium do? Well, the most important thing it does from a physiological standpoint is it allows for the specific and selective uptake of the nutrients that we digest from our meals. And in doing so, it also absorbs water and electrolytes such as sodium and chloride. It also secretes water and chloride ions to sort of fine-tune the fluidity of the contents of the intestine so that the processes of digestion and absorption can take place. And typically there's a balance between absorption and secretion such that there is a more prominent vector in the absorptive direction so that we're not losing fluid from the body. There are also other cell types within the epithelium that help to protect us from some of the pathogens, such as goblet cells that secrete mucus, cells called Paneth cells that secrete antimicrobial peptides. There are endocrine cells that secrete hormones in response to signals coming from the meal that regulate the function of all the other cell types. And the epithelium, in addition to having these sort of active functions, it's also a physical barrier. Even though it's only one cell layer thick, it does produce a fairly effective barrier that excludes toxic substances, it excludes pathogenic microorganisms, and the epithelium actually is also able to actively efflux certain toxins back into the lumen so that they're lost from the body. And then the other thing that's very characteristic, particularly of the intestinal epithelium, is that it maintains a lifelong relationship with a complex community of commensal microorganisms that live with us all the time. And some people have concluded that there are at least as many bacterial cells in our gut as there are cells in our body. And collectively, that complex mix of bacteria and indeed other microorganisms has a huge genetic repertoire with a gene pool that massively exceeds the number of genes that we have. And those commensal microorganisms are also very important for the biology of the gut. There is a sort of constant battle between commensals, which establish this sort of long-term relationship that is symbiotic, and potential pathogens, which have to establish themselves within the niche of the commensal microorganisms. But if they manage to do that, they can cause harm to the body. And a pathogen is simply a commensal that has acquired the ability to manipulate the host in some way to the bacteria's advantage. So for Salmonella specifically, it is able to actively invade into the intestinal epithelium, and by doing so can cause disease. The other thing that we have studied is the transport processes that allow for electrolyte absorption and secretion in the gut. And I'm not going to go into a huge amount of detail here in this sort of textbook figure, but suffice to say that there are specific proteins that allow for the movement of sodium ions and chloride ions either into the body that then drive an accompanying uptake of fluid, or in the secretory vector, there's movement of chloride ions from the body and out into the gut lumen, and that's how the intestinal contents are hydrated. And if we put this together in a sort of picture of a cross-section of the gut, in the small intestine, many of you probably know that the small intestine has these structures called villi. They look like little fingers waving into the lumen of the gut. That is really there as a device to massively increase the surface area of the epithelium. So that's how you get your tennis court. And that surface area is where typically we're absorbing nutrients in the small intestine. But again, there's this balance between the absorptive vector, which it mostly occurs across these villi, and the secretory vector of chloride ions, so that absorption normally predominates, but secretion is there as needed. In the large intestine or the colon, the structure is rather simpler. There are still crypts, but there are no villi. So, but there's a sort of analogous processes with absorption occurring across the surface epithelium. There is secretion occurring in the crypts. And the other thing that I should tell you is that the crypts are the site of the stem cells that populate the overall epithelium. And so the epithelium is kind of originates in the crypts and then migrates out onto the surface. And the epithelium in the gut is constantly turning over. Typically every few days in humans, the whole epithelium gets replaced. But as those epithelial cells migrate out from the crypts onto the surface or onto the villi and the small intestine, they change their properties and become absorptive. So in a variety of models, including a mouse model of Salmonella disease, which we developed along with collaborators, but also recently in so-called mini guts, where we can isolate the stem cells from the intestinal crypts, grow them in the presence of the growth factors that have been shown to be important for epithelial growth and differentiation. And we can either grow those in a sort of three-dimensional structure, which some people have called mini guts because it develops all of the cell types that's normally present in the epithelium, or we can kind of break open those three-dimensional structures and study the cells as a monolayer. We've used those models as well as others to identify that when mice or presumably people are infected with Salmonella, the bacteria invade into the epithelium, and it has two effects. One is that there is a decreased capacity for so-called electrogenic transport. So chloride secretion is reduced and sodium absorption is also reduced. Chloride secretion is a little paradoxical here because one thing I didn't tell you is that cholera occurs because that secretory process of chloride secretion gets turned on irreversibly. And so initially it was thought that that was what Salmonella would do as well. But in fact, we found that this chloride secretion is reduced in the setting of Salmonella infection. Instead, what appears to be the major mechanism driving diarrhea is sort of downregulation of sodium chloride absorption. And this then would prevent reuptake of the fluid that is used in the process of digestion or the fluid that we ingest with meals, and it remains in the lumen to cause diarrhea. And so this just shows this diagrammatically. There's a vastly reduced uptake of sodium and sodium chloride across the surface cells, but there's also reduced chloride secretion in the crypts. And this presumably has no impact on diarrheal symptoms. If anything, it would tend to mitigate the effects on absorption. But we think that it's very important for really impacting or negatively impacting the sterility of the crypts, such that you're no longer sort of flushing luminal components out from the crypts. And that might be deleterious to the stem cells and might cause a defective replenishment of the cells in the epithelium. So this is just sort of an example of the sorts of things that we're working on, trying to understand how bacterial invasion of the epithelium leads to mediators that are derived from the host epithelial cells themselves, appears to have effects on specific signaling events that control the differentiation of the epithelium, and or an effect on proliferation directly. And this then leads to a reduction in the expression of absorptive transporters and ultimately to diarrhea. So that's a really brief summary of my research interests. I'm just going to, in the last few minutes that we have available, talk a little bit about sort of what my career looks like in terms of what I spend my time doing. So as a professor, my major responsibilities are in research and teaching. So running my research group, teaching medical students, pharmacy students, and graduate students about my specialty. I actually taught for many years in the first year medical school curriculum. I would teach about GI physiology in general, and that actually encouraged me to write a textbook of GI physiology. A huge amount of what I do is mentoring. So not only the students and particularly graduate students, but also colleagues, junior faculty who are themselves trying to establish themselves in the field. I've done over the years a lot of service, and that has encompassed doing things for my university, but also outside the university. So I guess the time I'm spending now at NSF could be considered, you know, service on steroids, but I spend a lot of time contributing to scientific societies, various committees. It has been wonderful because I've got to meet people all over the world. I actually just this morning got an email from a colleague in Mozambique. I've had the honor of traveling to Mozambique twice now to advise the main medical school in the capital, Maputo, on how to build capacity in their graduate programs. And she was emailing me to say that they're getting ready to submit another grant proposal to continue to build out their graduate programs, and would I serve again on their advisory committee. So the last place I traveled out of the country before the pandemic was to Mozambique, and maybe that will be the first place I get to travel again. But the life of a biomedical scientist certainly gets you to meet people all over the place, and that's truly one of the huge honors of what I do. I mentioned earlier also that I'm really very much engaged, and this has been a consistent theme all my life, in the cause of diversity and inclusion in academia, in science, in medicine, wherever it might be. It's so important that we can call on the talents of everybody in this country and well beyond. We know that that when diverse sets of people come together with diverse backgrounds and diverse life experiences, they produce as a team much more robust solutions, whatever the problem might be, a societal problem, a scientific problem. There's a huge amount of research. So from a very pragmatic standpoint, we need a very diverse community of researchers and physicians and scientists and so forth. We also need all the people that we can get. In this country, we are going to have a shortage of scientists. And so we can't afford to be, excluding women or excluding members of underrepresented minority groups or anybody else who has the talents to contribute. And then, from my own, moral perspective, it's simply the right thing to do. Everybody should have these opportunities. And the other thing that is sort of an unexpected aspect of my career is I have had the opportunity to serve in a number of leadership roles, some of which I'll talk about. But as Violet Incredible, I never expected to be doing that. So I was asked to talk about what a typical day is. And this is my slide of a typical day. And this is to say that there is no typical day. And that's one of the most wonderful things about my career, is that on any given day, I could be doing any number of things. I could be, writing a paper. I could be working on a book chapter. I could be giving lectures. I could be talking to colleagues. At the NSF, I could be briefing members of Congress about the expected impact of proposed legislation, or how we can better help them to help the country move forward from a research standpoint. But there are two constants. There's always email, and at least for the last couple of years, there has always been Zoom. I want to encourage all of you to really think about how you can not only make steps in your to your destination of your career goal, but to think about the context and the institutions that you're working in. It was a real pleasure to speak to some of the leaders of the organization before we came online, and about how they got involved and what they were doing for the Pre-Health Chatting organization. But wherever you find yourself, I really urge you to get involved. There is such a reward to interacting with colleagues, to building your network, even for somebody as introverted as I was. I really forced myself to volunteer for things, and that has brought me so many opportunities. So you saw those links in the chat. This seems like a great organization to get involved with as a volunteer. And you may have many other opportunities in the setting that you're in. And then wherever you find yourself, think broadly about the organization, not just your little piece of it, but where are other places where you could apply your talents and could devote your time? And no matter where you find yourself, be prepared to work hard. But the rewards of doing things like this, of working hard, of stepping forward, of getting involved, it just gives you a chance to think more broadly and strategically. And as my career has developed over the, 36 years since I completed my training, I can see now, looking back, that all the things I pushed myself forward to do really built in a way that wasn't necessarily very planned or strategic, but it gave me the tools that I needed to move to the next step of my career. So having that ability to think broadly and strategically is really important. Whatever you end up doing, I really urge you to look for opportunities to make a difference, and maybe that's making a difference locally. Maybe that's making a difference to individual patients that you will interact with in the health professions. Maybe it's making a difference by discovering something that we don't yet know. And maybe it's making a difference by serving as a mentor or a trusted colleague. But you have the opportunity to build this network of colleagues locally, nationally, and internationally. And it's a chance to stretch yourself every single day. And that's the thing I like the most about what I do. You also get to do some really interesting additional things over and above what you think a professor might be doing. So I'm currently the editor of one of the major journals in physiology. I'm the first woman to ever be the editor-in-chief of that journal, and the first editor-in-chief based outside the UK, where the journal is published. That's a huge privilege because I get to see people's work before it actually hits the presses, and I get to have a hand in making it better and in giving people feedback to shape not only the actual written record, but the experiments that they report and the conclusions that they draw. I mentioned I authored a textbook of gastrointestinal physiology. I'm also the lead author on a comprehensive medical physiology textbook and a board review book driven from that. And then also I've contributed to many other textbooks aimed at medical students. I used to be a writer of questions for the board exam that medical students take, the US Medical Licensing Exam, Step 1. That was fantastic because for 4 years I would write 50 questions a year. It doesn't seem like very much, but it was actually really challenging. And we would go to Philadelphia for almost a week. And there was 8 of us on the committee. So we had 400 questions to go through. And I learned a huge amount. That committee was almost equally divided between clinicians and basic scientists. And since you're pre-health, you probably haven't really looked into the format of the USMLE exam, but it's very much written as a clinical vignette. So pretty much every question on the exam, even if they're designed to test basic science knowledge, starts with a patient. And so that was a wonderful opportunity for me to learn a lot more about clinical medicine than I did before. I was the president of one of my societies, the American Physiological Society, which was great fun to really sort of shape the future of the discipline, and came with some wonderful side benefits like trips to Brazil and Japan to interact with other physiological societies. Pre-pandemic and increasingly, hopefully, I'm a collector of frequent flyer miles. There definitely is a huge amount of travel to what I do, and that has been a source of just amazing experiences. I talked about Mozambique, but I have spent time in many, many countries and have met many wonderful colleagues from across the world. And then I'm also a backup singer in a scientific rock band made up of GI scientists. And that brings me to my sort of final take-home message. You must find a way to have balance in your life. It's incredibly important not only to focus on your career, but also to focus on the other things that make life important. Family, friends, children, if that's down the road in your future, doing things that bring you joy. And so singing in the band with my GI colleagues, almost all of whom are actually international GI researchers, is just fantastic. And we play at scientific conferences. We have had the opportunity to perform in various places in the US and in Canada and in London and in Dublin. And it's just so much fun. And this probably is one of the things I miss the most during the pandemic, is the opportunity to get together and play with the band. So with that, I will finish up, and I'm happy to take any questions.
Thank you so much for your presentation. It was really introspective, and we've learned a lot. So from your presentation, it is clear how you have a very reflective perspective on all of your experiences as a scientist, as a student, and as like a member of your team. So how has the biomedical sciences changed you as you went through your career? Like, do you view life and death differently, or has it changed anything in your personality?
That's a really good question. I think the biggest impact that my career has had on my personality is that I'm still hugely an introvert at heart, but I recognized early on that if I really wanted to make a contribution, I would just have to get over that. And like I said during the talk, fake it till you make it. I would go to conferences and in the beginning, I'd go to reception, I'd stand in the corner and I wouldn't talk to anybody. And now I'm sort of, from then on, I learned I just can't be that way and succeed. And a colleague of mine actually the other day said something that was hugely meaningful to me. And it really sort of resonates also with my love of mentoring, which is that he said, one of my strengths is to be able to talk to somebody about their research and get them excited about what they're doing and to see the important things. So no matter what you work on, it's important to care about the outcomes. And even though I am not directly taking care of patients, I really do believe that some of the things that we've discovered have down the road applications into actual patients who are suffering from these diseases. So I don't think at the beginning of my career I really thought about that. I was much more all about just the science and, oh, isn't this interesting? But it's at the end of the day, it's what it means in the real world that I care about now.
Yeah, of course, your mentor has taught you so well because your research has interested us so much. So thank you for presenting even as an introvert today. And so an audience member has asked, how are you able to balance your time so well, like between flying and talking to Congress, holding concerts, doing research? How do you balance all of that?
Well, I'm definitely a believer in the adage that when you want something done, you ask a busy person to do it. I've always been pretty organized, and I tend not to have too much in the way of downtime. I try to really, use my time as efficiently as possible. But I will admit, this is a very, very busy time at the National Science Foundation. In part because the presidential transition, everything about the budget got delayed. And so it's like all day long, I'm getting pinged with emails about questions about the budget and how do you sort of, how do we make the case for our programs? And it's been very difficult this week because I'm also supposed to be writing appraisals of all of my staff. And I sort of sit down and I open up the file and then another email comes in or an urgent message. And then in addition to that, the director of the National Science Foundation is going to Capitol Hill to testify next week. So as you can imagine, that takes a lot of preparation. He doesn't just show up and wing it. He actually has to be, it's like, I guess, preparing for the presidential debates. He actually sits in a room and they pretend to throw questions at him like it was in the hearing. So we had to provide a lot of materials with essentially a few hours notice for him, for his preparation. So it's easy to get derailed, but the one advantage I do have right now is that my husband is in California while I'm in Washington, D.C. So that gives me a little more leeway to do a little bit of work in the evening and on the weekend.
Yeah,
I get a lot of work done on planes. Sorry, I didn't mean to interrupt, but planes are a great place to get work done.
Sam, I do agree with that. And I do agree that the influx of emails can be stressful and it's really important to balance your time. Speaking of that, can you speak about your travels in other countries as a scientist? How was your experience like?
So I will say that the most wonderful thing about science is that it's pretty much, even though in different places people have different levels of resources available to them to do science, the sort of fundamental philosophy is the same. So wherever you go, you almost immediately have a bond with people. I guess, let's see, 3 or 4 years ago, I had the honor of going to speak at a conference in Nepal, in Kathmandu. And the reason I actually got involved in that is that one of the textbooks that I work on is the major textbook that's used for medical education in Nepal to teach physiology. And there was a junior faculty member at the medical college in Kathmandu who not only uses our textbook, but just was this massive fan of the textbook, and so had taken it upon himself to really read virtually every single line of that book, and would email me to ask questions, or would then email me to say, are you sure this is right on, page so-and-so and stuff like that. And it was fabulous. And he actually only has a master's degree because his institution doesn't offer a PhD in physiology. And but he basically stepped out there and said, I'm going to just send an email to this person who wrote this book. And so we basically, built up a correspondence. And then finally he said, I'm helping to organize this 3rd annual meeting of the Nepal Physiological Society. Would you come and speak? I had no idea how long it takes to get to Kathmandu. It's a very long way away. And I also had like a 12-hour layover on the way there. So it was a pretty arduous trip, but just getting there, meeting the students, meeting the people, a wonderful place. I urge you to travel there if you have the opportunity. It's absolutely beautiful. They were very much still recovering from the earthquake at that time, but rebuilding all the beautiful old temples and other buildings. But fundamentally, wherever you go in the world, if you are with other scientists, you will find common ground and things to talk about. And actually that faculty member from Nepal, I just found out last week actually that he is coming to the United States to do his PhD in physiology. He got accepted actually to 3 different programs. So I'm looking forward to seeing him in the US as well.
And hopefully you can see him and maybe collaborate on a research project. Who knows?
Yeah, that would be very cool.
And so with your dream of traveling, you've accomplished that. But why did you choose specifically to go to the US? Did you find it to be like a huge cultural difference that you had a difficult time adapting to?
It was absolutely terrifying. The saying at the time in the UK was that if you wanted to get a faculty position in the United Kingdom, you almost had to get a terminal degree, which they called the BTA, which stood for Been to America. And if you didn't have a BTA, it didn't matter if you had this great PhD or anything, but if you didn't have your BTA, you weren't going to get a job. And so a part of me felt that I had no choice but to come. But it was only maybe the 3rd time I'd ever been on a plane, and I arrived here with no money and 2 suitcases. I didn't know anybody. I came to work for an advisor that I'd never met. There wasn't even at that time email or Zoom or anything really. Even phone calls were really expensive. So I felt a very, very long way away from my family. The culture was extremely different than I'd grown up in the UK. I was a homebody to start with. The NIH was pretty terrifying because it had all these, as I mentioned, most of the people were MDs, which seemed to give them all already a sort of certain degree of confidence that I didn't have. And everybody just seemed so confident and to have everything together. Plus, everybody was quite a bit older than me because in the UK, undergraduate is only 3 years and I did my PhD in 3 years. So when I came for my postdoc, I was 24 and I was working with people who were physicians who were doing the fellowship. So most of them were, in their 30s and married, and they'd already pretty much established their lives. So I really did feel very much alone. if somebody had given me a plane ticket in the first 6 weeks, I probably would have gone home. But somehow or another, I stuck it out. I had a wonderful mentor, and I gradually got to know people and really appreciate the environment.
Thank you for sharing that. So as pre-health students, it's often a challenge to think in a broad manner and think strategically because we're in school and we just have this one like course or semester in mind. So do you have any advice for students who are struggling with thinking strategically about their career or personal development?
You definitely, it's really important to think about where you want to be and to sort of visualize yourself in that position. Not only because if you don't know where you're going, it's impossible to develop a roadmap to get there, but also when the going gets tough, if you can stay focused on the end result, then it helps you get through those very difficult and challenging times. It's not to say that you can't make course corrections as you go along, but I definitely have always encouraged trainees and other mentees that I've worked with. You know, you need your 5-year plan, you need your 10-year plan, you need to be able to see yourself where you're going. And quite frankly, as somebody who interviewed applicants for medical school and chaired the admissions committee for many years, I know at my institution now they've gone to a different interview format with these multiple mini interviews, but we used to have the sort of standard, 2 times 1 hour interviews with faculty members. And the first thing I would ask people is, if I meet you 10 years from now, where will you be? How will you be spending your time? What will you be doing? What specialty will you be in? And I'd also say, I'm not asking you to sign a contract. I'm not going to come and track you down and say, you said you were going to be a pediatrician. What do you mean you're doing orthopedic surgery? But you have to know where you're going. And it's really important to have those thoughts. And you can change it along the way, but at least have a sort of global picture of where you see yourself.
Well, someone in the audience does have a global picture. An audience member has asked, what advice do you have for students who are interested in research but are not sure where to start?
So it depends the environment that you find yourself in. If you're in a research university setting where there are plenty of faculty with research programs, you just got to knock on doors, basically, or at least metaphorically. Send out a bunch of emails, do your homework, get online, find out what people are working on. Be prepared to work in an area that doesn't necessarily seem like your first choice initially, because you're going to learn stuff wherever you go. If you're working in an institution where research isn't such a big part of the scenery, there are so many summer opportunities to get research experience. And so, look out for those, plan ahead, apply for them, and, be, brave enough to step outside of your comfort zone and spend a summer at another campus doing research. And the advantage of those opportunities is that they're often very structured, and they have a plan, and they will give you a lot of other activities, and very often you're part of a cohort, so you're with sort of like-minded individuals. But the advantage, of course, of doing research on your own campus is that you can typically do it for a longer period of time. You have to be pretty thick-skinned, because you'll knock on a lot of doors, and maybe people won't even answer your emails, but it's been my experience that people really do want to get involved in research and volunteer in a lab. They can usually find a way to do that.
Thank you. So Nanda had a question. She asks, what is your advice for students who are unsure about becoming a doctor or a scientist?
That's a really good question too. And I will say that if I was doing my life over again, probably in a different place in a different time, I probably would go back and do an MD PhD program, because while I've had the huge benefit of, working with colleagues who can really explain to me the direct implications for clinical work, I don't have the experience of treating patients myself. I do think I really have enjoyed the scientific part. If you're really not sure and MD, PhD seems like, too much to commit to up front, you can be a very viable researcher with the MD qualification, but you can never see patients with only a PhD, unless, of course, you're a clinical psychologist or some sort of special flavor of PhD, but a sort of regular STEM PhD, you're never actually going to be able to see patients. There are some advantages, though, to working in the field as a scientist, because you're not going to be, torn in many different directions and have somebody calling you from clinic about a prescription that needs refilling. And I do see that it is very challenging for my clinical colleagues. In general, I think we're equally busy, but I have much more control over my life and when I do things and how I organize my day.
Yeah, it is so important to be self-aware and to know what you want in a career. So yeah, that's something to definitely consider. But as someone who has had many years under their belt, who worked in the educational realm and the research realm, have you noticed like a shift in the research settings and the academics?
So the biggest shift I've seen is just the amazing advances in the field and what we can do and the tools that we can bring to bear. As you pointed out, I've been doing this for a lot longer than I'm sure all of you have been alive. And so I said, we didn't have email, we certainly didn't have Zoom. We couldn't have even imagined Zoom. Things took so long at the beginning of my career. Collaborations were so challenging because basically you had to write people letters and wait for them to come back. But then I felt so fortunate because, talking to some of the people who were my professors and mentors when I was an undergraduate and graduate student, they would come to conferences in America on a boat. They take a week to get here, and then they'd come and present their work at a conference, and then they get on another boat to go back again. So the fact that we can communicate so easily, the amazing, molecular tools that we have, the way that we have the ability to genetically engineer organisms, mice that express or don't express certain proteins, the molecular tools that we have to alter gene expression, the ways that we have to visualize things, even the sort of organoids, the mini guts I talked about. That's a technology that we've only really known about in the last 10 years or so. And you can actually get those stem cells, not just from animals, but you can get biopsies from patients and grow mini guts from their gut stem cells. And that just allows you the opportunity to really study the biological processes that you're interested in systems that are so much more faithfully reflective of actual human biology and disease. So yeah, just incredible advances. There are so many things that we can do. That's another great thing about being at the National Science Foundation. We get to see people proposing really exciting work, and they actually have a very robust program of scientists. They used to be actually coming in person to give talks, but now it's all on Zoom, but we get to hear from really some of the best people in the world.
Speaking on biological processes, Hannah asks, what is the difference between chemistry and medicinal chemistry? And what drew you to specifically medicinal chemistry?
So the biggest difference is that it's very much focused on sort of chemistry as applied to pharmaceuticals and drug design. And so in my medicinal chemistry course, I learned a lot of organic chemistry and some sort of basic physical chemistry. I really didn't learn any inorganic chemistry as a medicinal chemist, but it was supplemented with things like I took physiology with the medical students. I took a lot of pharmacology classes. So as I said, I thought I was going to be a synthetic organic chemist making drugs, synthesizing new agents for therapies, probably in the pharmaceutical industry. But it ended up being a great preparation for what I ended up doing, because I also had some of these the life science training along with the chemistry. And I don't use the chemistry part of my training, my undergraduate training, really anymore. But I still have the ability to sort of think in that sort of molecular way. And I think that's helped me in my career as well.
So it's really sad to wrap up, and it's hard to summarize like everything with just one last question, because we did learn a lot. But hopefully with people like going into the gastroenterology field, maybe they can present to you one day. And so with this, what future career goals do you have, and what advice do you have for others?
So my time as a graduate dean, my time here at the NSF has really cemented my desire to sort of contribute to an institution on a broad level. So I'm hopeful before the end of my career to have the opportunity to serve as like a vice president of a university or a provost, something that really takes the broad view and not just focused on my own research. That's been pretty much what I've done all along, but I'm really focused on that now.
All right, thank you so much for your presentation. Now we are going to be wrapping up. So give me a minute so I can share my screen.
So as we wrap up here, thank you so much again, Dr. Barrett. Is there any final advice that you'd like to share with us as pre-health students?
Just keep doing what you're doing. Stay focused on the prize. I think you all have a very bright future ahead of you.
Thank you so much, Dr. Barrett.
So students,
so
we're going to go into a quick wrap-up session. Dr. Barrett, you're welcome to leave the call if you'd like. Students, stick around. We're going to go into how we can get our hours here today verified. But yeah, thank you so much.
All right. Thank you. It was a pleasure to be here. Good evening.
Thank you.
Bye.
Bye. So I think we all learned a lot from our wonderful, wonderful speaker. So considering this, we would like to encourage everyone to reflect on the session today. What brought you to the session today? What are 3 major takeaways you got? And what do you want to learn more about? The writing or reflection part is not required. However, we do encourage you to submit it to our website for publication if you'd like for recognition. But it will be really nice if you want to keep a record of the sessions and keep a record of your thoughts and experiences. So if you want to learn more about pre-health shadowing and get involved in our program, we encourage you to visit our website. You can become an asynchronous volunteer to get certified hours through professional nominations, graphic design, and social media promotion. We are also accepting team member applications if you want to take on a more active role in PHS to lead projects, initiatives to be up here with us. Once again, we are humbly asking that if you're financially able to donate, that you please consider sending a few dollars to us via Venmo or PayPal. It costs a lot to keep our program up and running, and it costs a lot to keep it free for all of you because of Zoom and our website. So if you are someone who can afford to, or if you know someone else who can, please support those who cannot by donating to our organization so everyone can continue to get the education they deserve at pre-health shadowing. Otherwise, we simply ask you to spread the word about pre-health shadowing to reach as many students as possible. Now for the part we've all been waiting for, earning a digital certificate for our virtual shadowing hours in today's session. So first, you will be going onto our website and finding our professionals course page. In this case, we have Dr. Barrett. Next, you can take our quiz, which is a 10-minute quiz. It's multiple choice based on the content from the session today. You will have 30 minutes per attempt to earn a 70% or higher to get your certificate. We know that sometimes technology can be difficult, so for this reason, allow 2 attempts to take the assessment. But if you run into any difficulties, please contact us. To ensure that our website does not crash from a high influx of students, we recommend that you wait at least 30 minutes to an hour after the session to take the quiz. The quiz will be open indefinitely. So once you have passed the quiz at 70%, you can click the Finish Course button at the bottom of the professionals page and download your certificate, verifying your 2 virtual shadowing hours. If you happen to miss a part of the session or you want to go back to review, we do have our session recorded on our pre-health shadowing YouTube page. You can also find them via the professionals page in our website and take the post-shadowing assessments, which are also found on our website. Be sure to follow us on social media or subscribe to our email list for the latest updates on upcoming sessions and events that we have. We are currently booked every weekday through June for virtual shadowing sessions, so I hope to see you guys there. Thank you for joining us today, and please stick around if you've got any questions for myself or any other team members, and we will be happy to answer them for you. This shadowing session is officially over, and I invite you all to log off. Have a wonderful day.